Bismillahirrahmanirrahim. Assalamu alaikum. Hi student, this is Bukhar Ahmed and today we are here to discuss the different factors which help to control the microbial growth. This is our first lecture of microbiology and we move stepwise forward. First of all, introduction. Why? It is important to control the microbial growth. As we know, the microbes are the disease-causing agents. That's why they have ability to cause different infection in human body, and the personal safety is our priority whenever we are working in different labs, and in labs our body is directly exposed to different types of microbes which have ability to cause different infection. For example, if we discuss that the research shows that about 40% deaths caused due to typhoid fever are caused by the unsafe labs. Similarly, the Rocky Mountains fever also responsible for to cause more than 20% death, which are mainly caused due to unsafe lab techniques. And similarly, the hepatitis also have ability to cause different hepatitis is called different infections which are mainly transmitted when we are working with these viruses within the lab, and unsafe lab techniques in, unsafe lab techniques are mainly responsible for the transmission of such viruses in human body. And then third one, fourth one is the biological weapons. What means by biological weapon? Biological weapons is not directly related to the this one, but you must know what's mean by biological. Biological weapons are the weapons we are containing different microbes or other microorganisms, including both the viruses, bacteria, algae, fungi, etc. They have ability to cause different type of diseases, and even they have ability to cause the death of living of the humans as well as the other animals. Used These are used as a weapons and the wars in which such type of weapons are used known as the biological wars. Uh, Now we move toward our main topic, the methods used for the control of microorganisms. There are two main methods which we use to control the microbial growth. First one is the physical agents use, and second one is the chemical agents. But in this video lecture we are mainly focusing on the physical agents which are responsible for different microbial diseases. uh, Now move toward the physical agents used to control the growth of microorganisms. Physical agents mainly have ability to limits the growth of bacteria on non-living organisms. Main it's they in this type of mechanism we mainly use the alteration in temperature, ultraviolet rays, and filtration, etc., for the control of microorganism. And in the case of living organism or living body, the body we mostly does not use this type of mechanism. This type of mechanism mainly used for the killing of microbes from the different lab instrument equipments, etc. For example, surgical blades, the test tube, the battery dishes, etc., which that's used in lab in our daily routine. We They also have ability that they have microbes, etc. So to limits the growth of microbes in these equipments, we use physical control of microorganisms contain following factor. First of all, we discuss about the heat. Heat is the one of the best way to control the growth of microorganisms, because as we know that microorganisms are the living organisms. They have ability to survive in a certain limits of the heat, and if we exceed this heat elements. Then these heat elements have ability to denature the cell, denature their proteins, as the result of which the death of microbes occur. If we discuss about the heat, there are two main types of heat. First one is the moist heat, and second one is the dry heat. In moist heat, we kill the microbes by jarring and damaging the metabolic process. Mean the moist heat is mainly involved to stop the process of protein synthesis, while the dry heat is mainly involved in the denaturation or coagulating of the protein. Mean it's involved in the destruction or denaturation of the protein that are acting as an enzyme, that are acting as a as a structured protein. For example, they are forming the cell wall, cell membrane, or underspores, etc. In dry heat, we mainly focus on such protein. Now we in detail discussed about dry heat. A dry heat method was carried out in dry air oven. Or dry air oven mean these are specific oven in which we does not use any type of water, etc. etc. We just use the dry air, and the temperature of such oven varies from 160 to 180 degrees centigrade. This method mainly we used for the sterilization of the surgical surgical instruments, test tubes, petri dishes, fats, glycerol, etc. And we give time of 2 hours for the sterilization of such material. Second and most important method for, in the heat is the moist heat. The difference between the moist and dry heat is that in moist heat we use the water molecules, and in this type moist heat we use an instrument known as autoclave, and the process is also known as autoclaving. Inside this process we use water molecules, and the temperature ranges from 100 to 121. Mostly in moist heat or in autoclave we use 121 degrees centigrade for 15 minutes. And as we know, that's the boiling point of water is 100, and when we move increases our temperature from 100 degrees centigrade, our water is converted into water vapors, and these water vapors are mainly responsible to kill these microbes present inside the water. For examples, temperature below. These are some three types of heat, moist heat sterilization. First one is the temperature below 100 degrees centigrade, and second one is the temperature at 100 degrees centigrade, and third one is the temperature above 100 degrees centigrade, that is the autoclaving, and temperature at 100 degrees centigrade is the boiling, and the famous process of pasteurization occur at the temperature 100 degrees centigrade. These are the processes are mainly based on the nature of the substance which is sterilized, as we know mostly substances, when we increase the temperature of 100 degrees centigrade, they have ability to denature. That's why we use these fractions of temperature according to the nature of the instruments, or according to the nature of environment. Next and second method of the physical method for used for sterilization of material is the ultraviolet rays. Ultraviolet rays are powerful way of the sterilization, as we know that these rays have ability to cause different types of mutation. That's why these radiations have their applications only to the non-living organism, and mainly these rays are used for the cleaning of different lab. For example, gamma rays. We does not use ultraviolet rays for the sterilization of human body because these radiation have ability to penetrate inside the nucleic acid and DNA and cause different types of mutation. That's our today's lecture, and we discuss the other parts of physical resistance in our next lecture. Okay, thanks, and see my second lecture.